Welcome to the PharmGenEd Bridging the Gap Between Science and Practice Train-to-Trainer session for Toxicogenomics. Today is Wednesday, May 16, 2012. This presentation is supported by a grant by the Centers for Disease Control and Prevention. Its contents are solely the responsibility of the authors and do not necessarily represent the official views of CDC. The content is not intended to be a substitute for professional medical advice, diagnosis, or treatment. Today's agenda includes an overview of the PharmGenEd program, review of the educational content in the Toxicogenomics module, contact information, and a survey to assess program materials. There are webinars and additional therapeutic topics available on our website. You may also check there for updates about future webinars. The website will be provided at the end of the presentation. All copyrighted content included within this presentation has been granted copyright permission. No part of this presentation can be reproduced in any form without permission of the rights holder. The Pharmacogenomics Education Program, Bridging the Gap Between Science and Practice, is an evidence-based pharmacogenomics education program designed for pharmacists and physicians, pharmacy and medical students, and other healthcare professionals. The overall objective of the PharmGenEd program is to increase awareness about the validity and utility of pharmacogenomic tests and the potential implications of benefits and harms from use of these tests. These educational modules are currently available as part of the shared curriculum for faculty. At this time, I'd like to introduce Dr. Helen Smith for the Toxicogenomics module. Dr. Smith teaches in the Fike School of Pharmacy's Basic Pharmacology, Renal, and Oncology pharmacotherapeutic courses, as well as clinical toxicology classroom elective, a lab-based research elective in pharmacogenomics, and a lab-based APP elective in pharmacogenomics at University of Incarnate Word. Besides teaching, Dr. Smith serves on multiple committees, including serving as chair of the UIW-IRB. She has published in numerous peer-reviewed journals and most recently authored the Toxicogenomics chapter in the ASHP textbook. She has been awarded the UIW Faculty Development Endowment Research Fund, the UIW Faculty Development Grant Writing Summer Workshop Fund, and a research grant also with the Fike School of Pharmacy. She has a special interest in the development of toxicology and pharmacogenomics curriculum appropriate for pharmacy students and providing them with opportunities to gain research experience in pharmacogenomics. Dr. Helen Smith.
Hi. Thank you so much for joining us in this webinar today. Let me get to the slide here of our outline. Here we go. Today we will be following this outline for the webinar. First, I'll give a general introduction to toxicogenomics. Next, I'll discuss the current use of toxicogenomics in the context of occupational exposures, environmental exposures, and medication toxicity. I'll then close this presentation with a discussion of some emerging uses of toxicogenomics. The learning objectives of this presentation are the following. Upon completion of this program, participants will be able to discuss examples of genetic variants suspected to be associated with adverse effects of occupational exposures, and pertinent to this topic, participants will also be able to discuss the ethical and social issues that arise when genotyping is considered for workers. The next objective is the ability for participants to be able to discuss the current use of toxicogenomics and environmental exposures, and to discuss the use of genotyping to prevent medication toxicities. Let's begin this module with an introduction to toxicogenomics. The following are some definitions to distinguish toxicogenomics from pharmacogenomics. Pharmacogenomics is the study of how the genome influences patient responses to medications. Toxicogenomics is the study of how the genome influences the responses of those exposed to potentially toxic compounds. There is an intersection between pharmacogenomics and toxicogenomics, as these two fields of study share common approaches when studying the influence of the genome on response. Toxicogenomics developed from pharmacogenomics as the tools of pharmacogenomics began to be applied to toxicology questions. The concept of toxicogenomics has been a part of clinical medicine for some time, though it wasn't referred to by that name originally. One of the first applications of toxicogenomic concepts to medicine was the discovery that during the Korean War, soldiers of certain ethnic backgrounds developed severe hemolysis when given the anti-malarial Primaquine. It was eventually determined that deficiencies in the activity of the glucose 6-phosphate dehydrogenase, or G6PD, caused this susceptibility. Originally, G6PD activity in patient red blood cells was measured to predict the risk of this adverse response to Primaquine, whereas now genotyping can be done to determine this risk. The development of microarray technologies in the 1990s enabled high-throughput investigations of the effects of exposures to potential toxins on gene expression. This ability allowed the field of toxicogenomics to progress rapidly at that time. At this time, there are several applications of toxicogenomics that are being utilized in various settings, and a couple possible applications that as of yet are not fully utilized. For example, workers at risk for developing adverse health effects when exposed to select industrial compounds may be identified with genotyping. This application of toxicogenomics is used to some extent, but there are limitations to its use, which will be discussed later in slides. People at risk for developing adverse health effects due to environmental exposures theoretically may also be identified with genotyping. However, toxicogenomics is not yet being utilized in this context, but may in the future. The current limitations to this application of toxicogenomics is also discussed later in this presentation. Toxicogenomics is, however, being used as a tool in drug development and in clinical medicine. In drug development, toxicogenomics is used to investigate the mechanisms of toxicity and to predict the toxicity of medications. In clinical medicine, toxicogenomics is used to identify patients at risk for adverse drug reactions. Let's now talk more in depth about the use of toxicogenomics in the context of worker exposures. We'll start this discussion with the introduction of a case presented in the next slide. As we progress through the slides dealing with the use of toxicogenomics and occupational exposures, keep this case in mind and try to identify what the issues are with the patient presented. Here is patient case number 1. A 68-year-old male complains of shortness of breath, cough, chest discomfort, fatigue, and weight loss. This patient last held a job for several years machining beryllium metal parts. He has been retired for 3 years. The patient's symptoms have been worsening for quite some time. The patient consents, after talking with you, to participate in a case control study investigating the association between chronic beryllium disease, or CBD, and the genetic variant HLA-DP beta-1 glu69. Please note that the HLA-DP gene encodes a major histocompatibility complex class II antigen-presenting protein, and the beta-1 glu69 allele contains a glutamic acid residue at the 69th position of the beta chain. This polymorphism is thought to affect peptide binding to the HLA receptor. So keep this case in mind as we move further through our slides. Let's now talk about the toxicogenomics and occupational exposures. Genetic variations may predispose some workers to develop disease with workplace exposures. The following slides provide examples of genetic polymorphisms thought to influence the risk of disease or toxicity with certain occupational exposures. This list is not comprehensive, but it provides some information on known, potentially significant gene-environment interactions. The following toxic substances will be discussed: organophosphate pesticides and their toxicity, trichloroethylene exposure and the risk for hypersensitivity dermatitis, aromatic amine exposure and the risk for bladder cancer, and beryllium exposure and the risk for chronic beryllium disease. Let's start with organophosphates. Organophosphate insecticides are commonly used in the United States and abroad. They are responsible for a large number of pesticide poisonings due to their ability to inhibit cholinesterases, including acetylcholinesterase, or ACHE, and to some extent butyrylcholinesterase, or BCHE. The inhibition of cholinesterases results in the accumulation of acetylcholine, leading to muscle overstimulation. Some organophosphates, especially the more toxic oxon forms, are metabolized by the paraoxonase enzyme, which is encoded by the PON1 gene. Genetic variants in cholinesterases and in organophosphate metabolizing enzymes are being evaluated at this time for their association with the risk for OP toxicity. Two genetic variants, the R6-266-8207 and the RS-204-8493 in the butyrylcholinesterase gene, were found to be associated with decreased cholinesterase activity in this enzyme. These variants are currently being evaluated for their contribution to OP toxicity. It is suspected that inherently low activity of BCHE in agricultural workers may make them more at risk for OP toxicity. I just want to take a moment to point out to those looking at the notes portion of these slides that the RS in the mention of the two BCHE variants refers to reference SNP, and more information about these reference SNPs can be found in the NCBI repository database for the SNPs, and that website is given in the notes section of that slide. Now back to some genetic variants that are associated with OP toxicity. A second genetic variant of interest in predicting the risk of organophosphate toxicity is in the PON1 gene that encodes the paraoxonase enzyme. Data indicate that the Q variant of the PON1 protein is associated with a decreased ability to metabolize the oxon forms of organophosphates. The low activity variant of this PON1 enzyme is currently being evaluated as a predictor of sensitivity to organophosphates in a currently ongoing longitudinal study in Washington State agricultural workers. A low activity form of PON1 may allow for the accumulation of acetylcholine in exposed workers, contributing to an increased risk for organophosphate toxicity. The next slide begins our discussion of exposures to trichloroethylene. TCE is an industrial solvent and is a precursor for the production of other industrial compounds. It is known to cause several adverse effects with exposure. These include with topical TCE exposure, dermatitis. With inhalation and oral TCE exposures, workers can get CNS depression and renal and liver effects. And it is also suspected to be a human carcinogen and a possible teratogen. The incidence of TCE-induced hypersensitivity reactions is increasing, especially in Asian countries where this chemical is being widely used in emerging industries. These hypersensitivity reactions are manifesting as hypersensitivity dermatitis. Studies have found a significant association between the HLA-B star 1301 allele and TCE-induced hypersensitivity dermatitis in TCE-exposed workers. Please note that the HLA-B gene encodes for a MHC antigen-presenting protein. The HLA-B star 1301 variant is a 3-amino acid substitution in the MHC receptor, which data indicates affects peptide binding to the receptor. This change in peptide binding is suspected to play a role in the susceptibility of individuals for developing TCE-induced hypersensitivity dermatitis. Interestingly, the star 1301 variant has not been found in Caucasians or African Americans, but it has an allelic frequency of 10 to 13% in the Chinese population. At this time, the star 1301 allele is more common in the southern Chinese population compared to the northern Chinese population. The distribution of this allele in China may become less regionalized over time as people relocate to take advantage of job opportunities. Because of this relocation phenomenon, the HLA-B star 1301 variant may prove to be useful as a biomarker of those at risk for developing TCE-induced hypersensitivity dermatitis. This may be more important as the population relocates, as it will become less possible to predict who would be at risk for the disease based on their geographic location. Now let's discuss the risk for developing bladder cancer with aromatic amine exposures. Prior to the mid-1970s, aromatic amines were used in the production of aniline dyes. The use of aromatic amines in the production of these dyes in the U.S. is no longer allowed, but they may be used in limited capacity for research purposes and in industries in other parts of the world. Aromatic amines are known human bladder carcinogens. The N-acetyltransferases acetylate aromatic amines. The N-acetylation of aromatic amines is considered a detoxification reaction, preventing these compounds from undergoing N-oxidation that leads to the formation of potentially carcinogenic metabolites. A number of low-activity variants of N-acetyltransferases have been identified. The presence of these variants results in the slow acetylator phenotype. Studies of aromatic amine exposures have found an association between the risk of bladder cancer and the slow acetylator phenotype. The risk of developing bladder cancer has been found to be significant in the presence of the low-activity NAT2 star 5 variant in several studies. Now we will move on to talk about beryllium exposures in the workplace. Beryllium is another compound with potential for significant occupational exposures. Beryllium is used in many industries, including aerospace, ceramics, tool and die, nuclear defense, and prosthesis manufacturing. Some workers may be exposed to relatively high levels of beryllium in these work settings. Inhalation is the primary route of exposure to beryllium, with increased risk for workers whose jobs produce respirable particles, such as jobs that involve machining beryllium-containing products. Beryllium exposure can result in sensitization, which can be detected using an assay called the beryllium lymphocyte proliferation test. Beryllium sensitization may progress to the granulomatous pulmonary condition, chronic beryllium disease, or CBD. The rate of progression from sensitization to CBD is not known, but is thought to develop on average 6 to 10 years after exposure. It is also unknown whether all sensitized individuals eventually progress to CBD. CBD cases have been documented in workers exposed to allowable limits of beryllium in the workplace. The threshold of exposure for CBD is not clear. CBD susceptibility has been found to be associated with the HLA-DB beta 1 Glu69 allele of the MHC class II gene HLA-DP. We mentioned this allele earlier in our case that was presented. As I mentioned before, these alleles contain a glutamic acid residue at the 69th position of the beta chain of that gene. This polymorphism probably increases the risk of CBD by affecting peptide binding to the HLA receptor. We've just covered a few examples of some known genetic variants and their associations to workplace exposure disease. Now let's talk a bit more about the use of genetic testing in the workplace. Genetic testing of employees or potential employees is a possibility. The report by the American Medical Association's Council on Ethical and Judicial Affairs, published in 1991, noted the potential justifications employers may have for genetic testing in the workplace. These include an employer desire to identify individuals with a genetic predisposition for developing disabling diseases. This would save employers from the expense of hiring and training an individual who may not be able to work for long. A second justification of employers for genetic testing would be to inform employers which employees or potential employees will increase the health care cost to the employer. and the cost that the employer will have to bear for their employee. Third, genetic testing may provide information to employers indicating which employees or potential employees have a genetic predisposition that will cause them to be a public safety hazard in certain job descriptions. And finally, the last justification is that genetic testing could inform employers which workers or potential workers have a genetic predisposition for adverse effects from workplace exposures. Two types of genetic testing have been associated with occupational exposures can be done. These are genetic screening and genetic monitoring. They differ, and here are the definitions of those two. Genetic screening of individuals reveals specific inherited characteristics and may predict which workers are likely to develop workplace exposure-related diseases. Such genotyping data could be used to assign employees to jobs that would not expose them to the substances predicted to be harmful to them. The validity, however, of using genetic information in this manner is debatable. The second type of workplace genetic testing is genetic monitoring, and this may determine if a person's genetic makeup has changed over time because of occupational exposures. Besides indicating a worker has been exposed to a harmful substance, this data could be used to increase safety and health precautions in the workplace or change permissible exposure levels. The predictive value of disease risk with detected genetic changes is not yet well established, however. There are several ethical concerns with using genetic testing in the workplace. Genetic testing has added to our understanding of human disease, improving our ability to treat disease and to recognize those at risk for developing some diseases. However, although these data may be gathered with good intentions, they have the potential to be damaging to the employee and could be used unethically. The following are ways in which data from genetic testing in the workplace could be damaging or misused. Genetic data could be used to change, limit, or prevent employment. A worker's privacy could be compromised if this data ever got in the wrong hands. Data from genetic testing could reveal unanticipated heritable conditions that are not associated with workplace exposures. Knowledge of these heritable conditions could be damaging to the individual. Genetic data could also be used by insurance companies as a rationale for not insuring such an individual. These concerns hinge on the fact that there are unresolved questions regarding who has the right to the information available from genetic testing. There have been several instances when misuse of genetic data has occurred in the past. Also, many genetic tests at this time are not highly predictive of the development of disease or susceptibility to disease from workplace exposures. It is questionable whether decisions should be made regarding an employee's status based on the information that may not be highly predictive. Because of these potential ethical considerations or problems with the use of genetic testing in the workplace, efforts have been made to prevent discrimination and damage from the misuse of genetic information gathered in the workplace. The American Medical Association Council on Ethical and Judicial Affairs has taken the following positions regarding the use of genetic information gathered in the workplace. They state that it is currently not appropriate to exclude workers with a genetic risk of disease from workplace exposures. They also state that if genetic tests can be proven to be highly predictive, they may eventually be used in a limited way to protect workers from disease due to occupational exposures. Other efforts being made to restrict the use of genetic information gathered in the workplace stem from the Genetic Information Non-Discrimination Act, or GINA, passed in 2008. GINA prohibits employers from discriminating on the basis of genetic tests, reducing employment status on the basis of genetic tests, and requesting or demanding genetic tests of employees. GINA also inhibits insurance companies from reducing coverage or increasing pricing on the basis of genetic tests, and also requesting or demanding genetic tests from employees. Now that we have talked about some of the genetic variants known to be associated with workplace exposure hazards, and some of the ethical implications with using genetic testing in the workplace, let's discuss the first case that was presented earlier in this slide deck. Remember, we had a 68-year-old male complaining of worsening shortness of breath, cough, chest discomfort, fatigue, and weight loss. He had machined beryllium parts for several years prior to retiring. After enrollment into your study, which is investigating the association between chronic beryllium disease and the HLA-DP beta-glu-69 variant, he was genotyped for his status on that gene. As you evaluate this patient, you suspect he is suffering from chronic beryllium disease and will be categorized as a case in this study. You order a beryllium lymphocyte proliferation test to determine if he is sensitized to beryllium. This test does come back positive, indicating your patient is sensitized. Further clinical testing to rule out other causes of granulomatous lung disease confirm your diagnosis of CBD. The genetic testing that he has undertaken as a part of this study reveals that this patient is heterozygous for the HLA-DP beta-1-glu-69 allele. This would indicate to you that because of this allele, he has developed beryllium sensitivity, which has turned into chronic beryllium disease. Now let's move into our discussion of toxicogenomics in the context of environmental exposures. Let's begin this discussion also on the use of toxicogenomics in the context of environmental exposures by looking at another case. In this case, you have a patient that is a long-time smoker. She has a family history of smoking, but no family history of lung cancer. She has heard people can get their genes tested to find out if they will develop certain diseases. She is thinking of having her genes tested to see if she has an increased risk of developing lung cancer because she smokes. She wants your opinion of whether she should spend the money to get her genes tested. How would you advise this patient? As we go through the next several slides, keep this patient case in mind and look for information that would help you answer this patient's question. Genetic polymorphisms influence responses to environmental exposures as well as to occupational exposures. However, our understanding of these influences is limited. This is because of a few factors. These are that environmental exposures are complex, usually involving multiple exposures. The associations between genetic variants and adverse health effects from environmental exposures are not yet well understood. And another reason why we have a limited understanding of the influence of genetic variants on results of environmental exposures is that the adverse health effects resulting from these exposures will most likely be influenced by multiple genes. Efforts are ongoing to understand the influence of genetic variants on environmental exposures. One such effort is the Environmental Genome Project, or EGP. The Environmental Genome Project is an effort of the National Institute for Environmental Health Sciences. The long-term goal of this project is to characterize how specific human genetic variations or polymorphisms contribute to environmentally induced disease susceptibility. The EGP is conducted in 3 phases. The first phase is a resequencing of candidate genes to identify common genetic variants, and these genes include those involved in metabolism, DNA repair, cell cycle control, cell death, oxidative metabolism, signal transduction, and immune and inflammatory responses. This phase of the project is still currently ongoing. The next phase would be the investigation of the functional significance of polymorphisms in the coding and regulatory regions of these genes. And finally, the third phase of this EGP is the development of animal and cellular models to facilitate further understanding of the significance of the genes of interest and conducting population-based studies to understand how diseases resulting from environmental exposures are affected by these genetic variants. One of the more active areas of research in toxicogenomics and environmental exposures is the investigation of how genetic variants affect smokers' risks of developing lung cancer. Understanding these effects is complicated by the number of carcinogens in tobacco smoke and their metabolism. Tobacco smoke contains 30 to 50 known carcinogens that are metabolized by a variety of genes. Metabolized by a variety of enzymes. Several of these enzymes have been found to be polymorphic. Some of these polymorphic enzymes that may affect one's susceptibility to smoking-related lung cancer are cytochrome P450-1A1, CYP2E1, myeloperoxidase, and glutathione S-transferase M1. The impact of each of these variants on lung cancer risk in smokers is probably small. However, the total impact of these variants may be important collectively. At this point in time, however, the state of the science is such that a person's risk of developing smoking-associated lung cancer cannot be determined because of the limitations mentioned on this slide, or on the previous slides. Let me restate that. At this point in time, the state of the science is not allowing us to predict a person's risk of developing smoking-associated lung cancer and associating with these genomic variants because of the limitations of what we don't know about exposures and especially tobacco smoke metabolism. So now let's think back to our patient that talked to us about getting her genotyping done. She wanted your advice regarding whether she should be genotyped to determine if she has an increased risk of developing smoking-related lung cancer because of her genetic makeup. You inform your patient that scientists have identified variations in some genes that may influence the risk of developing lung cancer with smoking in some people. You also inform your patient that scientists have not identified all the genes involved and that we cannot definitively measure the risk of developing lung cancer with the data available at this time. You emphasize that, given what is known at this time, results from genetic testing would not accurately estimate the risk she has of developing lung cancer. You suggest that it would be much more cost-effective for her to quit smoking rather than taking these genetic tests. Now, let's talk about the use of toxicogenomics in preventing medication toxicities and understanding how some medication toxicities occur. Let's begin this discussion on the use of toxicogenomics for preventing medication toxicities by describing the situation in case 3, which is presented on this slide. Your patient is a 6-year-old girl found by her parents barely breathing in bed. The patient was rushed to the emergency department. A medical history was taken from the parents and a toxicology screen was ordered. The parents stated the child was being treated for a cold with a bad cough that was keeping her up at night. She had been prescribed a cough syrup with codeine. Data from a comprehensive toxicology screen identified codeine at the expected therapeutic concentrations, but morphine at elevated concentrations in her serum. Upon investigation, it was found the parents had been giving the cough suppressant properly as prescribed. What do you suspect is happening to cause this child to have respiratory depression at a normally prescribed dose of a cough syrup containing codeine? What test would you request to confirm your suspicion of what is happening with this patient? Now we'll move on to a discussion of the use of toxicogenomics in looking at medication toxicities. Many medications on the U.S. market have information in their drug labeling discussing the significance of genetic polymorphisms and their association with adverse effects. Such medications include, but are not limited to, abacavir, azathioprine, carbamazepine, and irinotecan. Polymorphisms have been identified that significantly increase the risk of severe adverse effects with the use of these medications. This table lists these medications with the potential to cause severe adverse effects in patients and the polymorphisms found to be associated with these adverse effects. Many of these drugs are already presented in other Pharm Gen Ed modules, but I quickly wanted to just point these out to you because of their relevance to toxicity. Abacavir, the first one on the list, has a boxed warning stating that serious hypersensitivity reactions have been associated with its use. patients carrying the HLA-B star 5701 allele are at high risk for developing this hypersensitivity. Azathioprine is, as you probably know, metabolized to 6-mercaptopurine or 6-MP, and then further metabolized to cytotoxic metabolites that can cause severe myelosuppression. 6-MP is inactivated in part by the polymorphic thiopurine S methyltransferase, or TPMT. Low activity variants of TPMT have been found, and these variants put patients on azathioprine at risk for myelosuppression since 6-MP is not quickly metabolized. Patients homozygous for the low activity variants of TPMT have a serious risk of developing life-threatening myelosuppression on conventional doses of azathioprine. Next in the table is carbamazepine. This drug can cause serious dermatologic reactions, including Stevens-Johnson syndrome and toxic epidermal necrolysis, or TEN. These adverse effects occur in certain populations of patients that carry the star 1502 variant. For irinotecan, it's metabolized to an active metabolite called SN38, and both irinotecan and its metabolite are cytotoxic. SN38 is glucuronidated by the polymorphic UDP glucuronyl transferase 1A1, or UGT1A1. Low activity variants of UGT1A1, especially the UGT1A1 star 28 allele, have been found to be associated with an increased risk of patients developing severe neutropenia while on irinotecan. So these are some medications with some severe potential toxicities, and as I mentioned before, the information on these genetic variants is included in their FDA-approved labeling. Patients are screened that are going to be put on these medications for the genetic variants mentioned above, and this is to identify patients at risk for these severe adverse reactions. Patients identified to be carrying these genetic variants may be started on lower doses or on other medications rather than the ones that they were thinking of using. Another way that toxicogenomics is being used, besides identifying patients potentially at risk for severe drug toxicities, is in trying to determine why an overdose situation might have occurred. Genetic testing currently at this time, I'm sorry, let me rephrase that. The use of toxicogenomics is thought to be potentially possible in determining why an overdose situation is occurring. However, at this time, genotyping may be beneficial. Genotyping cannot be done fast enough in an emergent situation to be useful. Therefore, the use of toxicogenomics in determining care in a suspected overdose situation is not currently routine. Furthermore, patients in emergent situations are being treated to remove the poisoning agent and prevent its pharmacological actions, or to treat the symptoms caused by the toxic level of the drugs. The clinical usefulness of genotyping information at this time is going to be probably limited in any emergent situation. However, though, forensics investigators will, in some cases, utilize patient genotype data to understand cases of acute toxicity. Genotyping patients after they experience drug toxicity can help determine why a patient's body burden of a drug was high. There have been a few documented cases of chronic drug administration that resulted in toxicity in the presence of certain genetic polymorphisms. It is possible that chronic drug administration may result in toxicity in the presence of certain genotypes. A few cases of apparent chronic toxicity in the presence of CYP2D6 poor metabolizers have been reported. CYP2D6 polymorphisms are often cited in the literature as possible causes of acute and chronic toxicity. This is understandable given the large number of drugs this enzyme metabolizes. Interestingly, in forensics investigations, there has sometimes been some discrepancies between a patient's phenotype and the genotype of a patient following a drug toxicity. There have been some reported cases in which drug levels measured in the body were not what would be predicted based on the patient's genotype. For example, Postmortem drug blood concentrations may differ from antemortem blood concentrations because of postmortem drug redistribution. This explains the discrepancy between genotype and phenotype. Another explanation for discrepancies seen between patient phenotype and genotype is when drug metabolizing enzymes with activating polymorphisms, there could be drug interactions that can inhibit the activity of the variant enzyme. This could cause the patient to appear phenotypically to be a poor metabolizer of the drug that has been displaced from the metabolizing enzyme. These discrepancies may impact the conclusions made in forensics investigations if the above factors are not considered. So this is an important twist in analyzing forensics data and understanding what your genotype is telling you in the presence of phenotypes. Finally, let's look at our summary of patient case 3. Based on what you learned in the last few slides on the use of toxicogenomics in the context of medication toxicities, you should be able to determine what is causing this child to have a respiratory depression at a normally prescribed dose of codeine. You suspect this patient has multiple functional copies of the 626 gene and is therefore an ultra-rapid metabolizer. This genotype would cause codeine to be bioactivated to morphine faster than normal, leading to an elevated level of morphine capable of causing respiratory depression. You request the patient's CYP2D6 genotype status to be determined. The genotype results indicate this patient is homozygous for the CYP2D6 ultra-rapid genotype. You suggest that administration of codeine in this patient be restricted. Finally, I would like to end this presentation with a discussion of the emerging uses of toxicogenomics. Efforts are being made to apply genomics data in emergency medicine in different situations. Providers are investigating the feasibility of using genomic information to diagnose emergent conditions. This may aid in diagnosing symptoms caused by adverse exposures. Keep in mind, this is the use of diagnosing an emergent situation and not to help determine treatment. Efforts are being made to use genomics information to improve drug safety in children. A national surveillance system has been established in Canada in an effort to improve drug safety in children. Investigators at the University of British Columbia have established the genotype-specific approaches to therapy in childhood, or GATC, Adverse Drug Reaction Network. Pediatricians who are part of the Canadian GATC surveillance network identify, enroll, and collect clinical data and biological samples from the adverse drug reactions cases that they see. At this point in time, the project focuses on collecting data from cases involving anthracycline-induced cardiotoxicity, cisplatin-induced hearing loss, and codeine-induced mortality in breastfed infants. The network is continuing to collect data and has proven to be an effective model of surveillance. So finally, I'd like to close this presentation by thanking you for your attendance. I'm happy to now entertain questions that you may have about this material at this time. Thank you very much.
Thank you, Dr. Smith. The references are also attached here. Okay. At this time, we'll continue the webinar by unmuting the audience, and we'll also go through some logistic information.
The conference has been unmuted.
So we hope this information is helpful as you translate pharmacogenomic evidence into practice. If you have any questions or comments, please feel free to email us at pharmacogenomics@ucsd.edu. Just like to thank all of our PharmGenet team here. This is also the post-training survey. Thank you for attending the webinar today. We greatly appreciate your feedback on this webinar. For those of you who are participants, the first link will be provided for you. For those of you who are faculty trainers in our Train to Trainer program, a different survey is available, and we'll send this email out with the appropriate links, which is used to kind of evaluate our program as well as evaluate our knowledge, attitudes, and self-efficacy of our trainers. So at this time, we'll open it up to question and answer session. And the first question from the chat is, what role might community pharmacists play in toxicogenomics healthcare? Dr. Smith?
I feel that community pharmacists definitely have a role in applying toxicogenomics in healthcare. For example, I would think that a community pharmacist should be checking to be sure that a patient has been genotyped if you're filling a prescription for abacavir, for instance. There's a couple other medications out there in community that community pharmacists might see, but this is probably the most important one at this point in time that pharmacists should be aware patients should be genotyped for before they are put on it.
Okay. A second question we have is, are you aware of any genetic testing or toxicogenomic studies related to Agent Orange exposures?
No, I'm not aware of those types of studies. I would suspect that they have been done. I wasn't looking for that. I haven't looked at that at this time. I'm trying to reach back in my memory. I think there were some studies being done looking to see about some of the oral vaccines that were used in the first Gulf War. And I know those were genomic studies, but I don't know what they were looking at specifically and what has been found with that.
Another question we have is, how much does it cost to conduct genotype 1 polymorphism, and how rapidly are the results available?
Well, I think that's going to depend on what method of genotyping you're going to use. There are some commercially available kits out there. Most of them that the public are aware of are the ones that are advertised as being able to genotype, especially your metabolizing enzymes. I'm not sure of the cost of those. And the results from those are pretty rapid. I think you mail those in and get those back.
Okay. And I think we have a hand raised, so maybe not. Does insurance pay for genotyping?
As far as I understand, genotyping cancer patients before they're put on irinotecan or for irinotecan, I think that is covered by insurance now, but I don't think genotyping is yet covered for abacavir. I can't completely answer that. I'd have to find that out for you.
Are there any questions just from the audience who are still on the webinar? Okay. I don't hear any questions. If there are no additional questions, we'd like to thank all attendees and Dr. Smith for the presentation. If any questions arise in the future, there is one question here. In order to use clinically, how much sensitivity and specificity is required? I'm assuming this is for the test, for the toxicogenomics.
Yeah, for the genotyping. I have seen, not for these 4 drugs that I use as examples, but I know that FDA does require certain specificity, or they require certain tests be done for some of the genotyping for drugs. I don't know what their parameters are for sensitivity and specificity.
Okay. I think there's another question from Dr. Williams.
Can you hear me?
Yes.
Oh, great. My question, I want to be very specific or be very knowledgeable about the specific genotype testing. Are we talking about testing for P450, the FDA-approved P450 test, when we're talking about abacavir and those 4 drugs you named? Or are we asking for another genotyping test?
No, if you go back to the slide where I had the table, it was slide 40.
Okay. Yeah.
Abacavir, for patients being put on abacavir, it's considered standard of care to test for the HLA-B star 5701. So the gene that will be tested for will be determined, based on what they're finding.
So you're genotyping from that particular variant. So the P451 that the FDA approved that I'm hearing so much about, is that only for the CYP2D6?
I'm not hearing the P450 being used in context with abacavir, but probably the CYP2D6 would be for things like the codeine. And it metabolizes so many other drugs too.
And Plavix, the one that is being on the commercials.
Oh, clopidogrel?
I think what you're talking about is the AmpliChip, the one that's
yes.
The AmpliChip is the one that's
talking about
approved.
The AmpliChip.
Right, for the Plavix and the warfarin testing, yes. And so those are specifically for what the AmpliChip would cover.
Well,
the cytochrome P450 I think.
Yeah, it'll depend again. Like Plavix is clopidogrel. Am I correct?
Yes.
That's going to be the CYP2C19, and for warfarin it should be the 2C9, but that's right.
We need to know. So so if I'm a nurse prescriber, if I'm trying to support my prescriptive decision making, I need to know the genetic variant that is metabolizing the particular med in order to know what genotyping tests I need to order for the patient.
Exactly.
And where's a quick and easy way for a nurse practitioner to access that kind of information?
One of the things that I teach my pharmacy students is to use the FDA's table of validated genomic biomarkers. You can get it off of the FDA website.
Okay.
And that table provides a list of all medications that have genetic information in their labeling. So if you were to open up a stock bottle of Coumadin and pull out that little insert label, that's the information that's in FDA-approved labeling.
So if I'm teaching nurse practitioners prescriptive authority and how to infuse this knowledge into their decision-making, prescriptive decision-making, they would need to have that table in front of them to know what genetic variant matches up to the drug that they're considering for that prescription.
Right. So that FDA table lists all the drugs that have genetic information in their labeling, and that labeling is that little insert you'd pull out of a stock bottle.
Yeah, I know.
Okay.
I know
yeah.
what the labeling is, but
right.
for for the practitioner in the field, collecting labels out of bottles.
Yeah.
I need a tool for the person in the field.
I would go to that FDA table, and what you can do is you access it on the web, and you can click on the links to the actual labeling, and that will give you the link to the language that you would need to decide which genotype you need to be testing for.
Thank you so much.
You're welcome.
Dr. Smith, can you repeat the name of the FDA table?
It's the FDA table of approved biomarkers is basically the name of the table. Dr. Lee, I think you're aware of the table. Is that the name?
I think you just Google FDA validated biomarkers.
Yeah.
Then I think it should come up.
Exactly. And they update that table quite often.
Good. We need to get the word out on that.
Yeah. I make my pharmacy students go through an exercise with it every year.
And I think somebody just posted on the chat function.
There we go. Yep, that's it. Thank you.
So, okay. Are there any other questions from the audience, either by chat or audio? PharmagyKB.org is also another good reference someone mentioned. Yes, and that's also on our resource page as well on our website if you haven't looked there already. There's also a lot of other links on our website that you can refer to. I don't see any other questions. So if there are no additional questions, I would like to thank all the attendees and Dr. Smith for the excellent presentation on toxicogenomics. If any questions arise in the future, please feel free to contact us at pharmacogenomics@ucsd.edu or visit our website at http://pharmacogenomics.ucsd.edu. Thank you very much.
Thank you. Please stand by.